Hello students. So today let us do this topic in reproduction. And let us first look at what we mean by asexual forms of reproduction. So first of all, any organism, its fundamental tenet is that of reproduction. So even if it is a virus which is neither considered to be living nor non-living, and yet its fundamental tenet is that its DNA needs to be replicated. So it will have to find out a host and within that host replicate as many number of times as possible. It's almost as if the organism is controlled by its DNA in order to reproduce several number of times. This is in fact the theory of Richard Dawkins. Coming to the two forms of reproduction, one form that we know of is asexual reproduction, the other form is sexual reproduction. Sexual reproduction is seen in vertebrates, and asexual form of reproduction is seen mostly in the lower invertebrates. Of course, it's not that sexual reproduction does not take place in invertebrates. It takes place in invertebrates also. But asexual form of reproduction is not seen by vertebrates. I mean, you won't get vertebrates reproducing asexually unless you consider parthenogenesis as a form of asexual reproduction. So this is one difference. The second difference is in terms of questioning why there is asexual and why there is sexual. So in sexual reproduction there is a formation of gametes and then they again reunite. Essentially this formation of these gametes, it ensures recombination, and this recombination is in a slow long-term method of achieving as much amount of variation. Whereas in asexual form of reproduction there is no gamete. So we have multiple clones. So these are actually almost as clones. Which are exact copies of the parent. So there is hardly any variation from generation to generation. And yet, because of mutation, it can give rise to a lot of variety. So although the variety that is obtained over here is little different and the variety that is obtained over here is different, but for instance, any virus can exist in a number of strains. We have been seeing in recent times that we have been affected by COVID-19, and you will see that there are variations, slight variations in strains in Europe, strains in Middle East, and the strains in India which are there. There are 2 or 3 varieties now. So how does that happen? That happens because of very quick mutations, slight changes and mutations within the clones. So it's possible even within clones to give rise to variations. So mutations take place here, but essentially a clone is just a copy of the original. Third thing is that this is very fast, and this is in terms of cost to the organism less expensive. This is slow process, and this is quite expensive. You should use, I am using the word expensive in the biological context, in the sense that it takes a lot of effort and energy for the organism to differentiate between male and female sexes. So we need to hear differentiation of sex. Into male and female, then formation of gametes, behavioral changes, morphological changes. It's very expensive for the organism. But here it's not expensive at all, it's very fast. So the reason of why this takes place is that this is basically to tide over difficult circumstances. Means that it can be a season which is a harsh winter or very difficult summer season. So asexual reproduction is very fast, very quick. Whereas over here, this takes place through all the seasons. So these are some of the differences between asexual and sexual. Let us also remember over here that there is nothing degenerate, or there is nothing of us something that we should consider a sexual form of reproduction as something being low and sexual reproduction being high. There are a large number of organisms that undertake asexual forms of reproduction for several millions and millions of years, and that is the way in which they survive. Take protozoans, for instance. Take the malarial parasite, for instance. Take ciliates, for instance. They all reproduce asexually. At least many of these, they reproduce asexually, and they are quite successful. And the fact that they are living even now, so they are quite successful. So asexual form of reproduction is also a valid form of reproduction. So now let us continue with asexual forms of reproduction. The first type is fission. Fission means to divide into two or more. So this is called fission, and in fission we will have one type which is called binary fission, and the other type is called as multiple fission. By the way, at this level in SYBSc, we are not going into a lot of details about this, an overview of the types of asexual reproduction. So, binary reproduction means two will be formed. Two daughter cells are formed, and here many daughter cells are formed. That's the difference between the two. So in binary fission, one parent divides and we give rise to two daughter cells. We see this in amoeba, for instance. So that is simple binary fission. So if this is an amoeba, it will have a nucleus, and this divides into two daughter cells. Now let's also focus over here and try to find out the chromosome number over here. So in the parent, the chromosome number will be 2n. In each of the daughter cells, this will also be 2n, this will also be 2n. This is going to be the pattern in all forms of asexual reproduction, that continuity has to be maintained. So essentially this is a form of mitosis. So this is diploid, this is also diploid. So an amoeba can easily divide into this. Let's also just focus over here is that preceding this form of division, the nucleus it disappears. That means you cannot recognize the nuclear membrane. But the chromosomes are there, and the chromosomes have doubled. This is preceding division. All the cell organelles they have also been resolved. That means within the cell you cannot see any of the cell organelle. Then it divides. And then there is restoration of all the cell organelles, and feeding again starts once again. So during this process of temporarily, when this division is taking place, feeding has stopped or it has slowed down substantially. After division there will be restoration, and then once again growth takes place. So this is, here you can see in the parent this is a little bigger, these two are a little smaller, then this will grow into the original size. This is what would be the same pattern in all forms of fission and all forms of, in fact, reproduction. So this is the parent and these are the two daughter cells that forms. So that is simple binary fission. But we can also say that in many forms there is something which is called as transverse binary fission and longitudinal binary fission. So for this is just the axis, okay, the axis of, mostly we will see this transverse binary fission in ciliate. In ciliate protozoans, and mostly we will see this longitudinal binary fission in flagellate protozoans. For example, if there is a this is paramecia. it will divide in this plane. Before this takes place, the nuclei have internally, although we cannot see it, the nuclei have actually doubled up. In many Paramecia there are 2 nuclei. In some there are more than 2 nuclei, that is, there could be 4 nuclei. It depends, but if you assume that there are 2 nuclei, in which the important nucleus is the one which is the smaller one, the micronucleus. The micronucleus has also doubled up, and that has been separated into 2. So there will be equality in distribution of all the nuclei, and this has given rise to two daughter cells, and ultimately this will also give rise to a protozoan, paramecium, and here also a daughter cell. Then again, once again the nuclei are restored. So here this will have, in this case, one meganucleus and one micronucleus, and the same thing will be seen over here as well. So let's look at the diagram for longitudinal is in, for example, Euglena. So this is Euglena. These are the 2 daughter cells which have been obtained. The plane of division is longitudinal. There can also be oblique type of fission, of binary fission. So this is A, B and C, and this of course was a simple binary fission. Transverse binary fission, longitudinal, and this we see, for example, also in flagellated protozoans. For example, in Serratium. So essentially the plane of division depends upon the morphological features of the organism, in which way it is organized, in that way, and therefore it occurs over here in a transverse manner, and in this it occurs in a longitudinal manner. Then we look at multiple fission. In multiple fission, if the nucleus repeatedly divides, without formation of daughter cells. Without the formation of daughter cells. So if this is the parent cell, within the same parent cell, without increasing in size, you will find very tiny structures like this, where the cytoplasm is also part of that. And if there was one nucleus over here, now multiple nuclei get formed over here. So here in multiple fission, the size of the original organism is the same. Here also and here also. Very quickly the nuclei are dividing. And what is happening is that the cytoplasm is apportioned. That means small portions of cytoplasm with nucleus. So this is almost like an amitotic type of division, because the nucleus is dividing very fast and without the formation of daughter cells. Within that same parent cell itself, small amounts of cytoplasm is associated or given to nucleus. This is called as to apportion. A portion of that cytoplasm is given to each nucleus. Therefore, it is called as apportioned. And therefore, from one to here, multiple is found. And after some time, this will break open to give rise to that is, the original cover has broken rise. Now, if these daughter cells are spores, if daughter cells they are spores, then the process is called as sporogony. And if the daughter cells are gametes, then this is called as gamogony. Both of these we see in the malarial parasite Plasmodium. So sometimes, and in different organisms. So these spores are given when, suppose there is winter season, and the animal will not be able to, or this organism will not be able to give rise to an organism at that time, because there is not enough food available. So it will give rise to spores. So spores are covered by a theca, by something that will not degenerate in that season, in that winter season. So we have formation of spores. In some cases, gametes are given or formed, male and female gametes, separately from separate daughter cells by that same process. So multiple fission can be called as sporogony also, it can be called as gamogony also. Another word which is often used is schizogony. The sporogony and schizogony are almost the same. Schizont means by division. So these are terms that different scientists have used at different periods of time. So this is all about fission. The next one is budding. Budding is also, you can think of it as a kind of an unequal formation. So if there is one, the best example is in Hydra. So if you show that this is Hydra, a tiny bud develops over here. And after some time, and when we say that the bud develops over here, budding, for example, in Hydra, and we have seen that Hydra will contain 2 germ layers. So when the bud is formed also, it will develop along with the 2 germ layers, and it will separate only when mouth is formed. So let's just look at a better diagram for Hydra. And only focusing on this part. So this is, let's say, the outer layer, ectoderm. So in the bud also, so this is the bud. So the bud will become bigger and bigger. So you can see that the same two layers, red and black, endoderm or endoderm and ectoderm, or epidermis and endodermis to be more precise, these two are also seen in the bud. The bud will keep enlarging. See, only the bud will become larger. And then at this level over here, there will be a constriction that appears. And after that constriction, there will be the formation of mouth and tentacles. Then separation. It means this so here the constriction goes on deepening. And now here you can see that the mouth has been developed with tentacles around it. Now it will separate. So it won't separate until feeding is possible for it. That's very logical. Otherwise, what's the point in forming a bud? The bud is not going to be able to feed. So it's a convenient method for organisms such as Hydra. It is also seen in many groups of annelids. So these are forms of asexual forms of reproduction, sometimes in coelenterates also. Other forms of coelenterates is a very, Hydra is a coelenterate, and other forms of coelenterates also. Budding is very common. Then we find a form of reproduction which is often called as fragmentation. So here we can talk about, say, for example, starfish, which is an echinoderm. Now, starfish is a predator. Just a little bit of an extra information over here. Starfish is a predator. It predates on oysters, and in many parts of the world, fishermen have got oyster beds and oyster farms, and the largest enemy of their oyster farm is a starfish. And if the farmer is annoyed with the starfish and breaks it, into pieces very quickly, each arm of the starfish will regenerate into an entire animal. So in fragmentation, a broken part of organism can regenerate into new organism. So as I said, if we are drawing over here a starfish, and let us say that it was broken up into these pieces, each one will give rise to 1, 2, 3 and 4. So you will get 4 starfishes, and this is by the process of regeneration. It was also classically studied in flatworms like planaria or even Hydra. In this planaria, if you cut the animal, so this will give rise to a planarium, this will give rise to a planarium, this will give rise to a planarium. And very logically speaking, A, B and C, A will be very fast, B will be very slow, and C will be the slowest. And interestingly, in the B part, this is still maintained as the anterior part, this is still be maintained as posterior part. That is the beauty of regeneration. So regeneration essentially involves de-differentiation followed by so these two concepts are important. When there is an adult organism, it has been differentiated. When it has to divide, there has to be de-differentiation. It has to come back to its original form. That's called as de-differentiation, and it must retain its potential to develop. So these two are very important developmental concepts that have been very useful in experiments of cloning, and later on they have been used in biotechnology. And then the fourth one is jabulation or formation of gemule. So this is also to tide over difficult circumstances. This is a form of an internal bud. For example, in porifera, in sponges, it's been seen in sponges. Now in sponges there are different types of cells, of which an important cell type is called as archaeocyte. The archaeocyte is a cell type which is important in reproduction. So archaeocyte is just a cell type. So a mass of these archaeocytes, they assemble together and they form a spherical structure, and now that is covered thick coat and protected by spicules. So the structure looks something like this. So this will be a pear-like structure. So these are the archaeocytes. This is the very thickening double coating, and in this we have got spicules. A spicule is a thorn-like structure. Of course, it can have different shapes, and it is made up of calcium carbonate. Spicule is a protective or defensive structure made up of calcium carbonate. So in this case, the shape of this looks like this. Each spicule is looking like this, elongated spindle-shaped structure with two concavities on either side. And here is an opening. Micropyle or opening. So this is a gemule. And this will remain for a couple of months without desiccation, without being spoiled. And when conditions become better, this is now rainy season is there, so this will open up, and so this is actually sealed over here. This part is sealed. It will absorb water, burst open, and then these archaeocytes give rise to a new several organisms. So this is called as Jabulation. So these are the essential forms of asexual types of reproduction. Thank you.